So I'm going to talk about animal locomotion, which, as Greg said, is my field of study. And if you're interested in the amazing things that animals can do when they move, this might be a good place to start. So these are videos that my colleagues kindly loaned me. At the top, we have a high-speed video of a fruit bat flying in a wind tunnel. Down here, we, of course, have a cheetah sprinting along. And these animals are just amazing by whatever measure you might choose in terms of their maneuverability, their stability, their efficiency, speed, power. And so it's not surprising that people interested in animal locomotion have studied these kind of animals for years. And it's also not surprising that roboticists, maybe very ambitious roboticists, people that want to make running or flying robots, have looked to these kind of animals for inspiration. And the success of being able to do that is increasing with every year. For me as a biologist, it's really fun to watch the progress in this area. And in fact, there's at least two groups that I know of that are working on a running cheetah and with the goal of getting their robotic cheetah to run at 50 or 60 miles an hour, which is about as fast as we think cheetahs can run. I've worked on animals like this, these animals that I would call they're sort of celebrity animal athletes in the past, and have really enjoyed it. But I've also worked with a lot of other animals. And today what I want to talk about is why sometimes we look to what you might consider unusual animal models to study locomotion and what they can tell us. So animals that you might not consider the sort of celebrity animal athletes. We're going to talk about the backbenchers today. And I know that one of the animals that I've worked on, Greg mentioned it, for 15 years or so, I know this from experience that when I tell people that I study locomotion in this animal, they really hesitate. And I'm pretty sure that this is the image that they get. So I study running in turkeys, and I have for a number of years. And I'm sorry to do this at dinner time. I know a lot of you haven't actually gotten to get to dinner yet. But the turkeys don't look like that in my lab. They look like this. This is high-speed video of a turkey running on a treadmill. We train these guys to run on a treadmill. In fact, they really train us to sort of give them whatever circumstances will allow them to perform really well. It's a treadmill that we just buy from a sporting goods store. This is a study where we're actually looking at downhill running in turkeys. So she's running downhill. She's used to it. She's looking around. She's checking things out. She's moving at a pretty good pace. This is slowed down quite a bit, so maybe you can't appreciate exactly how fast she's running, but she's probably doing about a 10-minute mile or so. So that's one of the animals that might be not the obvious choice for studying locomotion. I'm going to talk about some others tonight and sort of a brief outline about what I'm going to talk about. Why study unusual animals is a theme that's going to go throughout the talk. The benefits of curiosity-driven research. Research that isn't directed necessarily at an application, but can sometimes lead to important insights that have usefulness. Then I'm going to use sort of springs as a case study for what both robotic studies and animal studies can tell us about how springs can function in locomotion. And then finally, a slightly lighter topic, I'm going to talk about the challenges of working with animals that can do what they want, not always what you want them to do. So here's an animal that might not be your obvious choice when you think about locomotion. So if I say scallops and we did a free association, then a lot of you would probably say, maybe somebody would say wrapped in bacon, right? So you think scallops and you think of this, or you think of sort of the classic shell you can find on beaches just about anywhere, this beautiful scallop shell. But in nature, they look like this. They sit on the ocean bottom. There are really quite a few species. They sit there with these little filter feeding apparatus. These little blue things actually are little distributed eyes that they have all over the place. And they filter feed most of the time. Turns out that a really important study in animal locomotion was on scallops. So I want to talk about why we should study their locomotor behavior. And then if we should study it, should we mimic it? Should we make a robot that moves like a scallop? But before I get to that, probably a lot of you were thinking, what is the locomotor behavior of a scallop, right? Scallops do this, right? So I'm going to show you, and this is, so to get oriented, this is, so there are two valves in a scallop, two shells. There's a hinge back here at the back, and I'm going to call this side the gape side. And the way scallops swim is actually pretty cool. This is high-speed video from a colleague, Graham Askew at Leeds University, that's kindly shared. And it's going to loop, so I'll get you oriented here in a second. There's a scallop swimming across. There's a wire here because there's a transducer recording some important information about its swimming. I'll get to that in a minute. But here we're going to start. And it swims gape first, kind of smile first, with the hinge back here. It's really maybe not the way you would expect a scallop to swim. And it's really quite neat. It jet propels itself. It squeezes water out of little holes at the back near the hinge. You'll see the sand kick up when this thing swims. Poof. So it's swimming along with jet propulsion, which is pretty cool. But by a lot of measures, you'd say this is pretty lousy locomotion. It's not very smooth. It's herky-jerky. There's a lot of stopping and starting. There's actually a lot of, if we had another view, we'd see that the scallop is kind of rotating. So some of the engineers in the audience might say this is pretty wasteful locomotion. This is maybe not something we want to mimic. Why is it so crummy? Well, one of the points I'd like to make is that I think we often look to nature to sort of say what's the optimal design for something like a cheetah running. And you know, nature is not an optimizing process. Evolution results in things that work, but they only have to work as well as they need to. So scallops are crummy swimmers, but this is their predator. And this guy, this is a sea star. This is a fast sea star. He can go about 3 feet per minute. And scallops have to get out of their way, right? So all this scallop has to do is swim when the sea star comes. And really the locomotor task that the scallop has to perform is to be somewhere it wasn't at the end of the locomotor task, right? It doesn't have to do it fast. It doesn't even have to necessarily be in the right place. And if you think that's inefficient, that's how a clam moves. That's kind of cool. Right, so I would argue that scallops maybe aren't something you want to mimic. And they're an excellent example of how evolution produces designs that are adequate, not perfect. They're adequate for the task that has to be accomplished. So back to why you would want to study them. We want to understand how a complex locomotor and spectacular locomotor task like this can work. What we have going for us as physiologists is we know a lot about the motor that powers movement. We know a lot about how muscles work. And we know these from isolated muscle measurements, essentially taking muscle in essentially a test tube, electrically stimulating it. This would be a muscle, and historically this was frog muscles from dead frogs, electrically stimulating it and measuring the force and the power and the speed of the muscle. So we know from those kinds of experiments just how the muscle motor works. So what we'd like to be able to do is take that information and predict things like how fast somebody like this can run. The challenge with doing that, well, there are multiple ones. One is that the mechanics of running is really complicated. Multiple joints, lots of different forces, lots of different power outputs. Another challenge is that a runner like this is operating probably more than 50 different muscles involved in running. Each of those has multiple different individual motor units that power the movement. It's a really complex system. It's hard to take what we know about the motor and understand locomotor performance. That's where scallops come in, and this was recognized by Rich Marsh years ago, who was interested in these kind of questions. And one of the things he wanted to know was, actually, are these test tube measurements of how muscles work any good? There were some ideas that muscles operate really differently in a living organism than they do in these lab bench experiments. So he realized scallops were a great model. And we can see why, because here's our scallop again. Here's the valve. The hinge is back here. And if we do a cutaway, this is the tasty part that we like to wrap in bacon or saute. This is the muscle, and this is called the adductor muscle. This is the muscle that powers the closing of the scallop shells, that sends the jets back and allows the scallop to swim. So Rich used some clever device. So it's very simple, right? So we've got something that generates force. It's a single muscle, and you could make some assumptions that it's maximally active in a swim. So Rich used some clever devices in a scallop. He used these little things called sonomicrometer crystals. One of them sends out a ping of ultrasound. The other one receives it. And from the transit time and the known speed of sound, you can determine how far those things are apart. So he used those to figure out the length of the scallop muscle. And he used a pressure transducer to measure the pressure inside the scallop. And from known geometry, he could also calculate the force. And he could do other things that were neat. He took essentially tools from cardiac physiology, where people measure the work output of a heart and use the kind of approach where you know that the power output is the pressure that's developed and the change in volume within this area. So it's a great system. Single muscle, relatively simple mechanics, one joint, and he could ask these questions of how the performance of the muscle motor compares to the locomotor performance. Oh, sorry, that's a cutaway to show the contraction of the muscle. So the muscle contracts and closes the valve. So here's just, and so we learned quite a bit from these studies. I'm just highlighting two graphs, two of the things that we can take away from scallops. So on this side, we have measurements from a scallop during swimming. So this is a freely behaving animal. And this is the power output. This is essentially the horsepower that the muscle produces during a swim. And he did two different species, and there's some interesting things about species comparisons and temperature comparisons, things that we can learn that I'm not going to talk about right now. But one of the things you see from this graph, each of these data points represents a single clap from one of those swims that we saw. And one of these things we can see is that scallops get tired. So the power output is declining. This is an anaerobic activity. This is like an equivalent of us sprinting as fast as you can. You're going to get up to speed, and then within a few steps, you're going to start to slow down. And so swimming in scallops is pretty anaerobic, and that's actually not that surprising. It's actually anaerobic activity is fueled by glucose, right, by glycogen. And that's why scallops are so sweet, because they've got all this glycogen, all this glucose for these quick burst escape maneuvers that they do. So then the other piece of data is this. He actually took isolated scallop muscles, went to this test tube experiment I talked about, and moved them through the same pattern that he saw in vivo. So he did a different pattern. He did a sinusoid. I'm not really going to talk about that. But he did the natural rhythm of the muscle and measured its power output. And then he compared it with the in vivo, or what we saw during swimming in a freely behaving animal. And there's a little bit of difference here, but it really was quite close. And so we conclude two things from this. One is that test tube measurements of how muscles behave are pretty accurate for how things work in living animals, and we could move forward with that to understand other animals. And also that animals do seem to be able to get very close to maximal power output during motion. All right, moving on. I want to talk now, I'm going to tell a story that is sort of backwards in terms of how it was done. And I titled this Expecting the Unexpected, Why Curiosity-Driven Biological Research is Worthwhile. And I have to give apologies to Knut Schmidt-Nielsen, who used this phrase for a paper that made kind of a similar point about unusual animal models. There are a number of actuators out there that engineers have developed that are muscle-like. So they're muscle-like in that they're linear and that they shorten and produce force just like a muscle, as compared to a motor that's a rotary motor like you'd have in your car or any electrical motor. One of them is a McKibben actuator, and it's a pretty neat device. It works by having, here's an example of one. This is one that Greg Sawicki and Dan Ferris have used to actually actuate ankle motion to help people move their ankles. It's got a braided nylon sleeve on the outside, and then we can see in this cutaway it's got some elastic tubing. And what you do is you pump air into it. It's a pneumatic actuator. You pump air into it, and as you pump air into it, the elastic tubing has to expand. And because of the angle of the fibers in the braided nylon, as the diameter of this thing gets bigger, it has to get shorter. And it does this remarkably forcefully. And so people with various degrees of success have produced McKibben actuated robotic arms and robotic devices and assist devices. And we started playing with this. And so I'm an animal biologist, and this is maybe the first device I've made in a research context. And here's why we made a McKibben actuator. We think a lot about how muscles are put together. And muscles have architecture. So you might think about a muscle running from a tendon, which would attach to a bone, to another tendon that attaches to a bone. And between that, you have fibers that run along the line that the muscle operates in. A lot of muscles look like that, but a lot of muscles don't. In many muscles, you have the point where the muscle attaches to the bone, another point where it attaches. And rather than the force-producing fibers running straight along a line, they run at an angle to that line of action. And these are called pinnate muscles because they kind of look like the the barbs on a feather, or the pinny on a feather. So you've got fibers at an angle, and physiologists actually way back to the 17th century have recognized there's an advantage to that, and it allows you to, because it allows you to pack more force-producing fibers into this volume than if you arrange it in a parallel arrangement. So we made a pinnate McKibben actuator with, 5 actuators at an angle with a string down here to lift a load. And run that. Should be able to hear it. So what's cool about McKibbens is as they shorten, they also get fatter, and that's true for muscle too. All right, let me silence that for a second. That's true for muscle as well. Muscle fibers, as they shorten because they're mostly made of water and they can't change volume, have to get wider. So this thing lifts a load, and what you can see is these fibers are not only shortening, but they're also rotating, and they kind of have to do that just because of the bulging of these actuators to the side. And this is just a schematic diagram to show that. The fibers start out at this angle shown by the dotted lines, and then as these McKibben actuators shorten, they rotate. And what results from that is that you get an amplification of the movement of this load, of whatever you're lifting. So the amount that this load moves is going to be greater than the actual shortening of these actuators because you also have rotation. So you're going to get some sort of gearing, just like you'd have gearing on a bicycle, only we're not talking about something that rotates the system. And we found that sure enough that this system does give us some gearing that amplifies the movement of the actuator. But what was kind of exciting was that the gearing wasn't always the same. If we did a really light load, we got a gearing up above 2, meaning that we got a little more than twice the movement of the load than we got in the actuator along its length. If we increase the load, we made this thing lift something really heavy, we actually decrease the gear ratio. So if we have something heavy, we don't get as much movement here for a given amount of motion. So it would be just like changing gears on a bicycle. But this is happening absolutely automatically. We're not doing anything to the system. It just comes out of the behavior of this thing. So heavy loads, less rotation, and it's ultimately because of the way these things bulge and compress. Why did we do this? So we're animal biologists. Why are we making it? Well, we were inspired by mussels. And in fact, we were really interested in muscle bulging from the beginning, right? So everybody knows muscles bulge, and yet we haven't really known if that matters at all, other than for Mr. Universe competitions. And we looked at muscle bulging in turkey mussels, and what we found is that mussels don't always bulge the same. And I don't mean that my mussels don't bulge like the governor's, but they sometimes bulge in one direction and sometimes bulge in another direction. So we put these little sonar micrometer crystals, we measured the shortening of a muscle during a contraction, and then we used a muscle ergometer to measure, it's just a lever that measures movements, to measure how much the whole muscle changed length. And what we found was that we got the same variable gearing. So gearing, again, the amount that the whole muscle shortens compared to the movement of a single fiber. The muscle at low loads had a gearing of about almost 1.4, and at low loads the gearing was close to 1. And the reason was that the bulging, the way this thing changed shape, was different when you were at a high load than when you were at a low load. We think this is important. We think it's something like an automatic transmission that works within your muscles. It doesn't require input from the central nervous system. There's no nervous input. It just is an emergent behavior of how a muscle works. And despite the fact that we've known that muscles bulge forever, I don't think we had appreciated this before this. So why did we do that? Well, we did that, again telling the story backwards, because Beth Brainerd and Manny Anzizi had been really interested in salamanders. So a lot of salamanders are quite aquatic, and they swim kind of like a fish with lateral undulation. This is a siren salamander, a pretty big one. It's using little forelimbs, but it's also swimming using lateral undulation. And Beth and Manny were really interested in how they powered this. to tell the story relatively quickly. They looked at the muscles within the body wall that power swimming. And you might expect, as they did, that most of the muscles that are going to power that kind of bending are going to be oriented along the length of the body. And some of the muscles are, but there are multiple layers, and a lot of them are at an angle to the bending angle of this salamander. And they wondered why. They were simply curious about how salamanders worked. And through quite a bit of work and thinking and modeling, they realized that this rotation that I talked about that gives you some gearing, some amplification of the movement of the muscle might be important. It might actually allow an angled fiber to give you more bending and more swimming motion than a straight fiber. And sure enough, that's what they found. And so to kind of summarize quickly, this is the salamander. These are results of different models, making different assumptions about how the muscle bulges. So if the muscle bulges sort of in and out of the wall of the salamander, you get one result. And if it bulges sort of towards the back and towards the belly, you get a different result. And it makes a huge difference for a given amount of shortening of that muscle fiber, how much bending you get in the salamander. So they found that salamanders actually do something like this, kind of in between, something that gives them a lot of bending, not quite as much as the maximum, and not as much as if all the fibers just ran end to end. So the reason I wanted to tell this story was to demonstrate, I think, why. So we've made this device. We think that looking at muscles and their architecture in organisms as inspiration for how to build actuators for robots is worthwhile. Whether this specific device is useful or not, we'll see. But I think this is a good illustration of how purely curiosity-driven research, how does a salamander swim and why does its muscles look like that, can lead to thinking about a bigger problem like muscle bulging and why does it matter, and ultimately lead to a device. It's not as though we went out and said, how do we build a variable gearing pneumatic actuator? It came from the biology. All right, so moving on, this is a pretty spectacular video of some yellow-footed rock wallabies. It's loaned to me by Craig McGowan at the University of Idaho and work he did with Andy B. Wiener at Harvard. These are amazing animals by any account, again, some of our sort of celebrity animal athletes. And they're great examples of the importance of springs, biological springs in animal locomotion. And something like a wallaby or its relative, a kangaroo, we're talking about big springs in the limbs. And these guys have got these huge Achilles tendons, just like we do, but much bigger. And these Achilles tendons power this kind of hopping motion. This is one of the kind of showcase examples of the importance in springs in locomotion. And these guys are remarkably consistent. For anybody that's worked with animals, this is sort of, wow, it looks like it's looping. It is looping, but there are three animals doing kind of exactly the same thing. Everybody in the animal world who saw this was very jealous of Craig. So I've been interested in springs for a long time. And in my early days as a graduate student, we were trying to understand the importance of springs. And kangaroos and wallabies have springs. Here you can see the Achilles tendon spring really well. But we all do. Our tendons are stretchy. They're like rubber bands. They're quite stiff rubber bands, but they're stretchy. And that stretchiness is important because it can allow you to bounce off the ground like a kangaroo. But it's also, even for us, we bounce off of our tendon springs when we run. And we were really interested in the importance of this for the energetics of locomotion. How much energy does it cost to run? This was kind of the central question of comparative physiologists interested in locomotion when I was starting out as a graduate student. And it turns out that you can make measurements of how much energy it takes to move really relatively simply by just measuring how much oxygen someone consumes. So you may have actually seen this happen in a person running on a treadmill. You simply collect all the exhaled air, and the exhaled air is going to have a little less oxygen in it than the room air. And by the difference in content of oxygen between room air and exhaled air, you can figure out how much oxygen the person's using. If you're running fast, you're going to be using a lot. If you're running slow, you're going to be using less. So that's a pretty straightforward measurement. It doesn't matter whether you're burning fat or carbohydrate. It still works out. You can do it in a human. You can do it in something like a llama. You just can't buy the mask at a store. You have to adapt a milk jug, cut the end out, train the llama to run. If you have a small animal, you might just put him in a whole chamber and collect all the air out of the chamber. And this is really what we were focused on. And one of the things we're really interested in is looking at the pattern of energy consumption across size. So at one point, we actually took our show on the road and studied oxygen consumption in elephants. We did not have an elephant treadmill. We had to make this mobile. So the first key to studying elephants is to find an elephant trainer who is amenable to helping you out. And then we developed a manhole blower that we could put onto a golf cart to pull air through. The masks were made from 2 hefty trash cans. We used a little bicycle wheel and a bike speedometer to figure out how fast we're going. And then this guy had Von Langman, who actually organized this, had the most important job, which was to tell the driver of the golf cart if the elephant was getting too close. So we ran these guys through the zoo. We measured oxygen consumption. And that was an important piece of data on the sort of scale effects of locomotion. And the data I'm going to show now doesn't include the elephant data. But this is kind of a historical graph. This is way back from the 1970s from Dick Taylor, my PhD advisor. And it shows oxygen consumption, how fast you're consuming oxygen. Remember, this is essentially equivalent to how fast you're consuming energy. The graph's a little hard to see, so let me highlight 2 of the curves. On the bottom, we've got kind of a pretty big dog running along here. And energy costs increase as you run faster. That's not surprising. And up here, we've got a white mouse. And energy costs increases as you go faster. So there were 2 things that Dick Taylor and others wanted to understand from this graph. One is, why does the cost increase linearly? There could be lots of different functions. It might be curvilinear. That's actually what we see in birds, where a little increment in how fast you fly leads to a large increment in how much energy you use. But in running animals, it's linear. And then the other really obvious thing from this graph is that pound for pound, a mouse is consuming a lot more energy than a dog. And these other animals fall in between. And the reason they fall in between is not specifically in terms of, not because of how their limbs are built or any anatomical detail like that, but because small animals generally use a lot more energy than large animals pound for pound to move their body. So to move, if you put a little 1-gram backpack on a mouse, it's going to use a lot more energy to move that than if you put it on a medium-sized dog. So this is what springs were all about for us. We wondered what the role of springs was in explaining this. Since then, we've looked at the uses of springs in animal locomotion and found that really the tendon springs can do a lot of different things in running or in other activities. And I've sort of summarized that in a figure here. And the red is kind of meant to help you follow the flow of energy. So if we have our little stick figure and we think about running, what we think tendon springs can do in running is allow you to essentially act like a pogo stick or a bouncing ball, so that as you're coming in and you land, the energy you have a certain amount of kinetic and potential energy, and then you're going to decrease that as your leg compresses, and it's going to increase. You store that energy temporarily in this tendon spring, and then you release it. So that's kind of the one of the earliest sort of roles of tendons that we've appreciated. But there are other things that tendon can do. One is to actually take the work that a muscle does, temporarily store it in a tendon spring, and then release that energy rapidly to power movement of the body in something like a jump, and we'll see an example of that in a moment. And that we've seen in a number of organisms. Fleas use a mechanism like that with actually a catch mechanism that allows them to generate a lot of work, stretch not tendons but other elastic structures, and power these big jumps. And then another one that we've been interested in recently, and for those of you who've been at the meeting, Nikolai Kono has been demonstrating this really nicely. He talked about it on Monday, is that in fact tendons can also help us to get rid of energy. So if you're landing from a jump, the energy in the body can be sort of shuffled into the tendon initially as you hit, like a shock absorber, and then that energy can be slowly released to the muscle. And the tendon's providing different things in different cases. Here it's providing energy conservation, here it's providing power amplification from the muscle, and here it's providing what we've called power attenuation, or actually, which we think is important for reducing the risk of damaging muscles. It's like a shock absorber. Interestingly, you might expect that you'd see this in different animals, But we've seen all three of these actions, or these functions, in the same muscle in turkeys. So turkey muscle, a single muscle, does all three of these depending on the circumstances, depending on the locomotor activity of the animal. So I've had a lot of fun following what's going on in robotics. And in my early graduate days, I had the pleasure of getting to interact with some of the folks in the leg lab occasionally. And they were working on making robots that could run, a topic of the meeting here. And we went over to thinking, well, they're going to help us understand energy consumption in running. And we realized pretty quickly that if you're trying to get a robot to run, and you've been trying to come up with a control algorithm that'll allow it to take 2 steps without falling, and then maybe, the next PhD thesis, you'll get to 3 steps without falling, and so on. You don't really care how much energy it uses. You have to have a robot that won't fall down until you care about energy consumption. And it's been fun for me to watch, at least my view of the development, people can correct me if I'm wrong, is that when people did start adopting springs in use in robots, it really was about control. It was about this problem of moving without falling down. In animals, we had simply taken that for granted because they don't fall down, they just don't. We were just concerned with energy consumption. So now there are robots that use springs to help control, to help simplify the job of the person designing the algorithm that keeps the robot from falling down. There are some springs in robots, in robotic prosthetics actually, specifically Huher's very sophisticated ankle, that do something like power amplification, where springs allow the robot to overcome a limitation of what actuators can do. And I find it exciting that people are using springs to also try to make robots more energy efficient. As they become untethered, as they're not on a boom in a lab, people are, of course, more and more concerned with allowing the robot to be pretty efficient to move around. Okay, so frog jumping is a place where we found an important role of springs. This is a Cuban tree frog jumping from a force plate, which allows us to measure all the forces during jumping. Cuban tree frogs are actually around here. They're an invasive species, so they're in Florida, originally from Cuba and some of the other Caribbean islands. And these guys are pretty spectacular jumpers. This is a slowed down high-speed video. And we've been interested in these guys. This is a good example. This is a motion. We're interested in these just not because frogs are neat, but this is ultimately just an acceleration. It's a very rapid acceleration from an animal specialized to accelerate, to basically increase the speed of its body. But accelerations occur in all sorts of locomotion, even in walking. There's a little acceleration at the end of my push-off step. So this is kind of the extreme of acceleration, and we want to know what we can learn from it. And one of the things we can do with animals is broad comparative studies. And we did one recently looking at 3 different species of frogs. One of them was this sort of grumpy-looking guy, this cane toad here. These guys they're pretty big. They're actually also terribly invasive animals. They're these things that are eating their way across Australia after being introduced. This is sort of your common pond frog, a leopard frog. And then this kind of wrinkly Yoda-looking guy is the Cuban tree frog that you find around here. These guys secrete poison, pretty bad poison, from these specialized glands near their head. They don't have to worry a lot about predators, and they're actually not really great jumpers. These guys are pretty good jumpers, but they're pretty good swimmers as well, so we expect there's some kind of trade-off there. And then this guy, although he may not look it, the tree frogs are the really great jumpers. So we asked a really simple question recently, was if this guy can't jump so well and this guy's a spectacular jumper, we should be able to explain that difference from differences in their muscle motor. Ultimately, the power for a jump has to come from the muscle. So we measured the power output during a jump from the force plate that I showed you. And this is what we found. This is across time for a single jump, representative jump for each of these species. And this is the power output. This is the horsepower that the animal develops during a jump. And you can see that there's a pretty huge difference. So this essentially holds up our subjective judgment that these guys aren't very good jumpers, these guys are okay, and these guys are pretty spectacular. So we get really high power outputs for the cubans, in between for these guys, and quite low power outputs for the cane toad. So here's a graph that summarizes data for a number of animals now. And we've got the power output that I just showed you. And now it's indicated per unit muscle mass. So we basically know how much muscle is in their hind limbs available for jumping. And the crosshatched bars are the power their hind limb muscles can develop. And so there are two take-home messages from this graph. One is that you should notice that these guys generate a lot more power during a jump, but their muscles really aren't very different from the toads. The toads generate pretty measly power. These guys are kind of in between. There's no relationship between what the muscles can do and what the animals can do. So that gives you a hint that something beyond the muscles is really important. The second thing you should notice that also gives you a hint that something beyond the muscle is important is that these guys are generating a lot more power than their muscles can. So they're doing more than what their muscles are capable of. Both of these, we think, are indicators that there's a really important elastic mechanism. So these guys are essentially working like a slingshot. And what happens is you go from a toad with kind of weak locomotor ability to a tree frog that's a great jumper, is that you're not changing the muscle motor, you're changing the springs that it's connected to. So the basic model for what we think is happening is that you have a muscle, you have a whole bunch of muscles and limbs, but we can think about one as representative. It's connected to a springy tendon. It contracts slowly, and this is good because muscles generate more force and do more work when they're slow. And then the tendon recoils really fast. So if we look at the power of a jumping frog across time, early on, there's very little power developed if you measure it by the movement of the body. We think during this period, the muscle's contracting to stretch the spring. During this period, when power is high and actually exceeds what the muscle can do, you're getting that slingshot effect. So we were able to test that directly. Henry Astley, a graduate student in my lab, used a newly developed technology at Brown, which involves Xray measurements of either bone or, in this case, radioopaque markers. So he implanted very tiny, about the size of a poppy seed markers within the muscle fibers. So the frog has this big ankle extensor with a long tendon, kind of like the Achilles tendon that goes around the ankle. So if we implant these markers, we can see what the muscle does and relate that to the motion of the ankle. Excuse me. The animal's anesthetized for this. These are tiny markers. It doesn't cause it discomfort. It recovers and jumps and performs well. So here's X-ray video of a frog jumping. You can see the little radioopaque markers. Here's the frog, and he's jumping. And actually, this is a pretty awful jump. Somebody pointed out to me that jumps should go up and not down. But we did a whole bunch, and sometimes the frog jumped really well, and sometimes it didn't. I'm going to show you data from one of these not very impressive jumps, which I think actually makes it even more convincing. The little shadow you see here is just a latex glove on a stick that we use to sort of wave above the frog and get it to jump. And the data look like this. So we use this new technology that's been developed at Brown called XROM, where we not only use the X-rays, but we add to that a CT model of the bone. And we can recreate an incredibly accurate model of how these bones move in space during a locomotor activity. So this shows the bones of the ankle, and you can see the little radioopaque markers that we're using to track the muscle. And then this is a graph. So this shows the motion of the ankle down here versus time. This is where at the point in time where the frog jumps. What you can see is the frog sitting, 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 and then you get this very rapid extension of the ankle. That's when the motion happens. But what tells us that the spring-like mechanism is happening is that before there's ankle motion, you get a lot of shortening of the muscle. So what's happening is the muscle fibers that are in series with this springy tendon are shortening, just like in the schematic I showed you, stretching that tendon spring, just like a slingshot. And then you get this very rapid motion of the ankle that's powered partly by muscle shortening, but also largely by that tendon spring. So I found it really exciting. Peter Adamczyk showed a a model of a little robot that they built at Michigan that explains maybe how this loading and release occurs. How do you basically load the muscle without the frog taking off early on in that period? And I can talk about that in questions if you like. So in the last little bit, I'd like to talk about, just give you some insight into what it's like to work with animals and some of the challenges. And so, as I said, one of the things I've appreciated watching roboticists is that it's very hard to get a robot to walk around or run around without falling over. People are doing a pretty spectacular job of it now. But essentially, every step a robot takes, you have to tell it how to do it. You have to tell it how to do it without falling over, how to do it to move forward. You are the CNS. You're the central nervous system. You're the brain. And so to me, that seems very challenging, right? But at the same time, a challenge for animal physiologists, we don't have to tell the animal exactly how to put one foot in front of the other. But it's also a challenge that we're not the CNS. So we don't have to tell an animal how to walk, but if it doesn't want to walk, and we want to study walking, that might be a challenge. So if I want to study jumping, and this frog would prefer to mug for the camera and sit in my hand, there's a problem there. And so this problem was really highlighted in a recent project we did. And that project was essentially trying to deal with a really simple question, which is how far can a bullfrog jump? Simple question, maybe not completely on its own terribly important. But it turns out bullfrogs have been an important model for locomotion for a long time. And this is data from 4 different studies that each estimated maximal jumping performance in a bullfrog. And they're a little bit variable. But they're all kind of converging on, I don't know, about a meter. So a bullfrog can jump about a meter. And people did various different things in these studies to try to determine that they were getting maximum performance, what a bullfrog can actually do, not just what it wants to do. And I was part of some of these studies. And they're challenging experiments, but I felt pretty good about them. The way we motivate these animals to jump is just to sort of verbally encourage them, or sometimes just to wait, just to sort of, put a cloth over them and lift and hope they get sort of startled and jump. There are lots of different techniques, but we feel like with some experience, we get pretty good at it. So the problem that came around was that it actually came from this book of my son's, the Guinness Book of World Records. It tells us how far frogs jump. There's actually a record for a sharp-nosed frog, but then they mentioned that at the annual Calaveras Jumping Jubilee held at Angels Camp, California, an American bullfrog called Rosie the Riveter, owned and trained by Lee UGC of Santa Clara, California, leapt 21 feet 5 3/4 inches back in May 1986. And when I read that, we were sort of floored when we saw that. Now that's sort of here to the wall, right? That's a really long jump. So we panicked for a little bit. Then we found out that, in fact, the way this contest is run is that it's the distance for 3 jumps. So we breathed a little sigh of relief. But then we did some math, divided that by 3, and realized that we still had a problem. Rosie the Riveter, if you assume each jump was equal distance, is jumping out here greater than 2 meters. And all the scientists said that bullfrogs can jump about a meter. This bugged me for about 10 years before I finally decided we had to go find out what was going on. So in reality, the arrogant scientists, I thought, well, these guys don't, they don't know how to measure how far a frog jumps. I've got to go see what's going on. So this contest is an amazing institution. It's I think it's in its 85th or 86th year this year. It was inspired, of course, by the wonderful story, one of Mark Twain's earliest stories about a gambler who jumped a frog and put buckshot on another guy's frog to win a bet. Anyway, a great little story. And it occurs in gold rush country in California. And so in the 30s, this area, as sort of the gold rush was waning, instituted this frog jumping contest to recognize this legacy and probably to sort of get a little bit going. It happens at a county fair. So this is the stage. This is the frog jump stage. This is where the contestants are. Here's some strange scientists that have come to study it. And there's a little launch pad right there. And actually, you can see the plaque that says Rosie the Riveter has the world record. This happens to be the world record holder jumping. And you can get a pretty good idea how this works here. So there's a launch pad. That's where the frog starts. These are the teams in waiting. And he's going to have the frog jump. Stickmen, like any good institution, it has its own jargon. So stickmen are guys that come out, see a little wet spot where the third jump ended, put the stick down, and then people come out with a tape measure and measure how far that frog jumped. Now, what we didn't appreciate when we got into this was just how serious this contest was. This goes on for 4 days. The contestants, are largely families that have done this, sometimes for generations, and are really serious about winning the contest. This, for example, is in the staging area, various coolers. This is a box. It's got various different chambers that are then subdivided with many frogs in each box. The teams come to the contest often with specialized trailers with temperature-controlled flowing water, frogs within it, and they're there to win. And the best way to appreciate that is to look at some pictures. So here's one of the teams. This guy's obviously very invested in how well this frog's going to do. This guy's really determined to motivate the frog. There's a scorekeeper in the back. There's a net guy who's ready. So this is a hard contest because appreciate not only does a frog have to jump really well, but it's a straight line distance of the 3 points. So if your frog jumps 7 feet and then 7 feet and then 7 feet, you've got a 0-foot jump even though your frog was amazing. So great facial hair is not obligatory, but is encouraged. And then, I mean, if you've seen an athlete concentrating, this is what you're looking at here. So here's somebody, and you can see that they don't just put this frog down and let it go. They are with the frog. I mean, it's spectacular. And we pretty quickly, after we got there, realized we dropped the arrogant scientist stuff. And realized that these frogs were doing pretty spectacular things. And this is probably my favorite photo. This is my "You Will Believe a Frog Can Fly" photo. I don't even know where he's took off. So when we were trying to arrange this trip, I contacted the frog committee that arranges this, and we got to know them later. And she actually told me that when we first called and said there were scientists that wanted to fly out from the East Coast to study their contest, she thought it was a hoax. But eventually, they were just wonderful to work with. But in one of the early conversations, I was sort of starting to explain, well, you know what I'd like to do is get all of these frogs and jump them off my force plate. And at the time, I didn't understand just how serious this contest was. And she was sort of said, wait a minute, you're not going to touch these frogs that people are competing with. And she said, but we do have rental frogs. and my reply to that was naturally, rental frogs. I'm not familiar with the concept of rental frogs. So they have rental frogs. So they have actually a few hundred frogs that they actually get from the same river drainage, the source of these other frogs. You can, for $6, earn a frog and get a ribbon. We all did it and jumped our frogs. And actually, if your frog does well, you're right in the competition with all the serious, what they call professionals. So we were hoping to jump these rental frogs on force plates and just handle them, and we were going to watch the contest, but we didn't think it really mattered. But we pretty quickly realized that when we handled these frogs, the rental frogs, we were getting jumps that just were like just like what we got in the lab, which was not very good. So fortunately for me, my student Henry Astley had planned ahead, and we actually ran out to the Ace hardware store, bought a bunch of PVC pipe. He had already looked into a MATLAB algorithm to use a de-distortion algorithm to calibrate the stage. We put a camera up in the stands. So with the PVC pipe calibration grid on the stage, which they were kind enough to let us do, we could get about a 2-centimeter accuracy of position on the stage, which would allow us to get jump distance of these frogs. And we recorded video for 3.5 days during the contest. So in the kind of work that we do, we're usually dealing with N of 3 or 4 animals. We try to minimize the use of animals, and it's also challenging work. Here we were able to get a huge number of animals. And as I said, there's its own language. So the people that come with their own frogs are called professionals. People like us that rent frogs are civilians. The person jumping the frog is a frog jockey. So we got data for somewhere around 2,600 jumps for professionals and 600 jumps for civilians. And just to give you a little high-speed video to give you some idea what it looks like, I actually find it a little more entertaining to watch the jockeys than the frogs on these. We ended up not doing a lot with the high-speed video. And so I can show you what we got with just a histogram, a distribution. So this is the jump distance in centimeters, and this is the number of jumps we got. So for rentals, for civilians, for people who paid their $6 jump frogs, we got what you might expect, a normal distribution. Some of the frogs jumped really well, but not very many. Some of the frogs, I think my frog was probably here, didn't jump much at all. And then there was kind of a mean. But one of the things we learned just from this is that so the scientists are in trouble already. So the very best jump in any of the scientific literature is out here at a 1.3 meters, I think. And some of the jumps, if you jump enough frogs, are a little bit beyond that. But what gets interesting is when we add the professionals. So first of all, it's not a normal distribution. It's very skewed to the right, and most of the jumps are better than what we thought a bullfrog could do, and some of them are truly spectacular. We can compare this normal distribution for the rentals with the professionals. And I think this skewed distribution is really starting to tell us that we're running up into a physiological limit of what the bullfrogs can do. Here, what you really have is a lot of behavioral noise. The challenges of a central nervous system. So one of the most interesting pieces of data actually came from a free newspaper when we were having lunch during this research excursion. And that was the winning jump distance, going all the way back to the 1930s. And what I see here is a pretty clear trend, which is generally, with some noise, winning jump distances were increasing up until about the 70s or 80s. And then you hit what is a plateau. So the message I take away with this, and there are completely viable alternative explanations, is that it took hundreds, if not thousands of people jumping frogs over 50 years to figure out how to do everything just right to get to what was truly the physiological limit of what a bullfrog can do. And I can talk a little bit about what it is that allows them to really jump frogs well. I think if you want to ask questions about it when I'm at the end, but I think you could divide it into brains versus brawn. Are these super frogs, or are they just well-motivated frogs that are behaving well? I think it's more the latter, but there are arguments to be made either way. I think it's an interesting open question still. So in conclusion, I just wanted to talk about why study animal locomotion. These are actually a couple of my boys at the contest. One of them, I think, is a little more natural frog jumper. The other one's not quite so sure. Of course, they're brothers, right? So they're rivals. So he's still really sore that that Will's frog jumped about 5 feet and Ollie's hardly jumped. But anyway, why study animal locomotion? So I think it does give us insight and inspiration for device design. And we've seen, for those of us at the meeting, we've seen lots of examples of that this week. I think looking to biology for inspiration is a great idea. I think it's effective. And I think sometimes it comes from looking at animals that you think can tell you how to make a given device, and sometimes you get surprises, as we saw with the variable gearing McKibben muscles. So the other thing that motivates my research, I didn't talk about it a lot here, but the muscles I study have really common properties, whether you're talking about a frog or a mouse or an elephant or a human. And certainly the physics that governs locomotion also is conservative. And so you can learn basic fundamental features about how the musculoskeletal system works from studying animals, and that can be relevant to problems of health and disease, and that is certainly something that gets me out of bed in the morning. But there's another one that I think is important, for me anyway, I feel like is an important reason to do these kind of things. And this is that applications are great, but not everything worth knowing has an application. I want to attribute that to Roger Cram. That's where I heard it first. I've actually asked him what the original source of the quote is, and I haven't heard back. But I think that's a good point. I mean, the way I like to think about it is that I'd like to think that if our kids ask us, well, how is it that, how do frogs jump so far? The answer we'd give them is, that's a great question. Let's find out. So I'd just like to give thanks to people in my lab and elsewhere, to funding agencies, and to the animals. Thanks. So I assume there's time for questions. I can take questions, Greg?
So why not do Frankenfrog experiments where you replace the nervous system with a massive electric shock and just totally deplete the whole ATP and everything?
Well, so one of the really fun things about talking about this calaveras experience is that it depends on the audience you talk to. So one of the fun things is figuring out why these guys jump so far. So the geneticists all say, oh, I know, they're breeding special frogs. And the behaviorists say, oh, they're training them. And so I was wondering what the engineers would come up with. Taking it a little more seriously, the data suggested, I mean, if you massively stimulate the muscles, you don't get anything more than what the nervous system can do. So they're an isolated muscle with maximal stimulus. That's one of the things the Scallops study tells us, is that the nervous system can get you right there. You don't get anything more or less from a person with a big shock than you do with the nervous system doing what it ordinarily does. That's a more legitimate. Other questions? Yeah.
Do you feel like the frogs jumped farther in this Calaveras County competition because there was a different environment than, say, in a laboratory? Do you think that maybe they were more comfortable in a lab?
So that's a good question. So one of the things is the lab. So at least one of the scientific studies was done sort of out in the wild with animals that were just caught. And that's actually the one that has the longest jump. So that one, I think the animals would be, presumably just as comfortable as they are in a stage in the cold country of California. But that's a reasonable concern. I think that what we saw is that the frog jockeys who'd been doing this for a number of years, comparing notes, they're all very secretive about what they do and their techniques. But they all had a bunch of very stereotyped things they would do. If you saw how people were holding the frogs, they all sort of did that the same way. Of course, they all pursued the frogs. They actually had this quote that I think I would have thought was silly until I saw the whole thing happen, which was that the frog senses the will of the jockey. Which I would paraphrase as saying, they know that we're not really going to eat them in the lab, that we're just trying to motivate them. So I think it's all these kind of subjective things. Sometimes in the lab we get frogs from a supply where they're not freshly out of the wild, and so probably that has some effect, and that it is an artificial environment. Yeah?
In the two histogram, you measured the length, but have you tried to measure, for example, the weight or other things on the frogs and on the individual frogs and try to relate them? So have you found something in that direction?
Do you mean for the Calaveras frogs? The contest frogs?
Groups, for example,
oh, yeah.
you measure something because then it's just a number, but the individual frogs are not the same.
Yeah. So one possible simple explanation would be just that the professional frogs just had bigger hind limbs, for example, and we just didn't have the access to be able to get that kind of data. Again, subjectively, I didn't feel. What we did we started doing it by little froggy mug shots. We would just hold up frogs and take pictures and then measure the diameter of the legs. And but it didn't look like there were differences. The professional jumpers have their favorite fishing holes that they catch these frogs in. But overall, the rentals were coming from the same river drainage, so the same general area. Whether there were super frogs that had a physiological difference that we simply didn't measure, I can't say yes or no. I didn't. But I didn't, from sort of carefully watching, I didn't see obvious differences in these frogs. And the really good frogs weren't necessarily all the same size. Some of them were a little bit on the big side. Some of them were a little bit on the little side.
Question for the cheat-sheets. You studied turkey locomotion.
Yes.
You didn't talk too much about it in the talk. What's the most surprising thing you discovered studying turkey locomotion?
I think one of the early discoveries was probably most surprising, and that was just how important these tendon springs are. So we found when turkeys are running and they hit the ground and their ankle flexes and extends like ours does, we used to think that some of that was done by the tendon stretching and some of it was done by the muscle, but we found for that muscle at least, that can be done almost solely by the tendon spring. So the muscle's just generating force statically and the spring is doing the rest.
And was that one of the first times that was really
So
that extent?
it was one of the first times it was seen directly in a muscle. That was something that with turkeys we could actually see what was happening at the level of an individual muscle. And in other cases, we'd done it with kind of black box experiments.
Thanks.
So thanks, good question. Other questions? But yeah.
Yeah, so I noticed earlier on your technique for getting a frog to jump was to wave a glove around. And from looking at the photographs of the people out in California, they seem to adopt a bit more aggressive strategy that involves some form of hand waving, possibly ground slapping, and pursuit of the frog. So could you comment on maybe what differences the technique for inducing the jump might have had on this?
I think the technique makes a huge difference. You saw those pictures, and they were not, we don't look anything like that in the lab when we're getting frogs to jump, as you pointed out. We try to do pretty well. The X-ray is kind of a special case because whereas I might, come up right behind a frog and go, bah, to get it to jump, sometimes they just look at you. You can't really do that in the X-ray because it's X-rays, and it's not enough X-rays to harm the animal, but you don't want to get in there, so you're using things on sticks, so you don't get fantastic performance. But back to the sort of general idea, yeah, we obviously learned that we had some learning to do about how to motivate animals. And, yeah, up there.
I'm thinking of the image of a diver on a diving board.
Mm-hmm.
And I'm wondering what application you might find for introducing energy at the point of surface resistance where they're springing from something. Could you add spring underneath the spring? Go ahead.
Yeah, it's a great question. So one of the interesting things about these tree frogs is that they're jumping from tree branches. And so they're jumping from probably springy branches already. So a good question would be, can they use that to their advantage and get jumps that are even better? It's something we haven't really looked at in detail. We have actually had these guys jump off some springy perches, and what we found was that their performance didn't get worse, but it didn't get better. And they're not getting worse is kind of interesting because they have to jump in the right way that once you put energy into the springy branch, you want to try to get it back, and it seems that they're pretty good at doing that. Yeah, back here.
So when you look back at the papers that underestimated frog jumps, with your new knowledge, frogs can actually jump way farther, how does that affect the conclusions of the paper?
Yeah, that's a great question too. So, in science we like to sort of set up these straw man and show, so-and-so said this. So in this case I could point at myself and my collaborator Rich Marsh, we basically had gone back in previous, you can find in print, where we said, well, these pond-like frogs like bullfrogs have to swim as well as jump. And so there are trade-offs associated with swimming, and that's why their performance is so poor. That argument gets a lot weaker now, because their performance, if you take really good jumps, gets closer to the tree frogs that look really good. So that's one place. The other place is that this elastic mechanism, we saw it operating in these submaximal jumps, so now we know it works in the pond frogs. But previously, the power amplification, the difference between the power of a jump and the power from the muscle was so small in those frogs that we didn't have that much confidence in the elastic mechanism, and now we have a lot more confidence. So those are two places where it's changed. Yeah, question here.
Do you think if you got the frog jockeys to motivate the superstar frog jumpers, that they would go further as well? Or?
You mean the other species? Maybe, although I suspect some of the motivation stuff is going to be species specific. So that's a good question. Interesting thing about the contest is there isn't a species rule. So you can bring whatever frog you want, but it has to fit within the 4-inch round circle that's the starting point. So years ago somebody brought Goliath frogs, which are these huge African frogs, and made a big stir, and actually turned out one of them didn't fit on the circle. One of them did, but the day of the contest it was too hot for those frogs to do well. But It's interesting, the tree frogs, we feel like their performance is a lot more regular. We feel like the behavioral component, we're just subjectively, again, working with these animals, they're just a lot more stereotyped in their behavior. They usually jump about the same distance. They usually jump pretty well. Why there's a species difference, I don't know.
About the scallop, could you say something about, say, the control loop? Because that's the muscle. What the sensors and the processing that it gives is, how is it biologically?
Well, that's a good question. The place where I feel like we have some insight into that, so it's not a cyclical activity. So in some ways you could think it could be really simple, feed forward, just stimulate all the muscles, right? But they steer a little bit and they vary in how much they jump. But the one thing I think we got that was kind of an interesting piece of interesting little window into one of the challenges of operating a springy system is that I don't know if you noticed in the graph of the bullfrogs, but we saw what we consider backfire. So after they loaded that spring, it's like a bow, right? And once you load the bow, you want to release all that energy to the arrow, but you could release it back to your arm. Same thing with a frog. Once the muscle stretches the spring, you want all that spring energy to go to the kinetic energy of the body, and we see backfire. So we see places where the muscle gets stretched, even though the ankle is extending. And that's, I think, a control challenge of a muscle spring, muscle tendon actuator that we haven't fully appreciated before.
But I mean, how does it sense and what is processing of the scallop? So how does it feel that the predator is there? What's
Oh, that, for the frogs?
no, for the scallop?
Oh, the scallops, yeah, they've got those little eyes. That's one of them. So they've got, yeah, so the eyes mostly, I think they sense light getting blocked and some motion. So they can't make out shapes, but they sense the motion and they move. Yeah. I'm sorry, I thought you were asking about frogs.
One last question.
Okay, we'll come full circle.
So does aerodynamics matter? Are good jumpers actually good flyers? Like flying squirrels?
Oh, you mean generally, not just frogs, but all around?
Well, let's focus on the frogs. They look like ski jumpers to me.
Yeah, so it look like they do something, and short answer would be I don't know. It's an open question. Yeah, it definitely looks like they do something with their limbs. Thank you.